As we know, there are many conserved patterns in the sequences of DNA. As we have seen earlier, that before the gene starts, there is a region called the promoter region. And in this promoter region, there are many conserved sequences called motifs. So we can find these motifs by using our specialized techniques. So in this module, we will try to find out how we can figure out the exact location of the motifs within the DNA sequences. Here we can see the figure number 1, and we can see 7 sequences of DNA. And all these sequences are composed of 32 nucleotides. So we can see in the next slide, here what we have done is we have added a conserved region known as P pattern within the 7 nucleotides of these patterns. So the sequence of the pattern is ATGCAACT. So we can see here in the first sequence, we have inserted this pattern in this location, in the second in this. So at randomly we have added this pattern within all the 7 sequences of DNAs. So we know the location of this pattern within the 7 nucleotides of DNA. But the problem is, if we do not know the location of this pattern, so how we can find this within the sequences of DNA? So the answer is very simple. We will simply count. We will see in the next slide. Here we can see the same pattern is present within the 7 nucleotide sequences, but now we cannot recognize, they are not underlined the patterns, and we cannot see any pattern within these 7 nucleotides, although they are present within these 7 nucleotide sequences. So how we can find these patterns that we have incorporated within the sequences of DNA? So the simple answer is that we will count the number of times each elmer or string of the length occurs in the sample. And we also know that the total size of the 7 strings is 280, because 7 are the number of DNAs, plus 32 is the number of the nucleotides within the each sequence, and plus 8, this 8 is the number of nucleotides, we are calling it a pattern, that is present within the each DNA sequence. So the total, if we calculate it, 7 multiplied 30 plus is equal to 7 plus multiplied by 40. So the answer will be 280. So from here to here, the total number of sequences are 280. So the chances, probability is very low that any of the 8-mer sequence will repeat more than once, except for this pattern for which we have incorporated within these 7 nucleotides. So we will count the pattern of 8 mers, 8 nucleotides one by one, and we'll find that only this pattern will appear 7 times. So the probability of this 8-mer pattern, other patterns to appear more than once is almost negligible. So we will come up with the 7 nucleotides of this pattern sequence. Again, we can see that in actual DNA sequences, the pattern, this one will not be conserved. So in the actual problems, here we can see in the first nucleotide sequence, this is the pattern that we have incorporated, and we can see that at A, T, it should be G here, but here it is C. And in the 6th element, it is G here, it should be A. So in this way, we can see that in this pattern, there are 2 changes in the conserved region of P in the first sequence of nucleotides. Similarly, in the second DNA sequence, here is the conserved pattern, but here again we can see there are 2 changes in the first and second element. it should be A or A and T, but instead of A and T here it is G and G. The rest of the pattern is identical. So that is the case for all of the patterns that we have incorporated within the DNA sequences. So in the real-time problem, in the DNA molecules, there is a tendency of mutation. So due to the process of mutation, these nucleotides, they often change with other nucleotides, and this conserved pattern could not be conserved as we can say here P is equal to A, T, G, C, A, C, T. So there are many mutations within this pattern. So after mutation, there is a difficulty to find out this pattern. So to cover up this or overcome this problem, we design a specific technique called as matrix. So by using the techniques of matrix, we can overcome this problem of mutations within the P pattern within the sequences of DNA. So we have seen if we do not know the exact sequence and the location of specific pattern within a DNA molecule, we can, and we know that if there is also a phenomena of mutation within the patterns, so we can find the consensus sequence of that pattern by using the technique of alignment matrices. So we will see this alignment matrices in detail in the next modules.